Hi student, I am Dr. Sagar Kumar, MSc PhD, and I am having 32 years experience in the teaching field. They were the 11th class and 12th class, including the NEET exams also. Today I am going to teach in this video aerobic respiration. Aerobic respiration means in the presence of the oxygen, the respiration used to take place. And in the aerobic respiration, it will be done in the mitochondria. Aerobic respiration will be taken place in the mitochondria. In this aerobic respiration, glucose will be stepwise completely oxidized. Oxidation means releasing of the energy, completely oxidized. And next in the mitochondria and releases large amount of the energy and large amount of energy with water and carbon dioxide. This is the brief about the complete oxidation. Means in the mitochondria, glucose will be completely oxidized stepwise and releases the energy with the water and carbon dioxide. So this will start with pyruvic acid. Pyruvic acid, why? Glucose is to forms two molecules of the pyruvic acid in the glycolysis, all of you know. Those pyruvic acid will be transported from the cytosol to the mitochondria. In the textbook also given cytosol. What is the cytosol means? The cytoplasm without cell organelles, entire fluid is called as cytosol. Note this point, remember it properly. And from the cytosol to the pyruvic acids are transported into the mitochondria. And in the mitochondria, two crucial steps will be taken. One step will be taken in the matrix of the mitochondria. What is the matrix? The fluid which is to present inside the mitochondria. And the second step will be taken in the inner membrane of the mitochondria, because mitochondria covers with two membranes. Inner membrane of the mitochondria, in the inner membrane of the mitochondria, the second step will be taken place. What are those steps? In the first step, complete oxidation of the pyruvic acid stepwise and releases the hydrogen, means hydrogen carbons are present in the pyruvic acid. So pyruvic acid, whatever the hydrogens are there, they will be released and forms the NADH plus H plus like this, and FADH2 hydrogen, and ATP also used to form in this. And next, what it will do, where it will be taken place? In the matrix. In the matrix, what will happen? Complete oxidation of the pyruvic acid will be taken place and produces the NADH plus and FADH2 and ATP by releasing of the hydrogens and carbon as the carbon dioxide. Means whatever the components are present in the pyruvic acid, all will be released. Whenever they are releasing, it is the catabolic, that is oxidation. So complete oxidation will be taken place in the matrix. Now the second step is, in the first step, FADH2 and NADH plus H2 H plus are produced at all. These will be provide electrons, and these electrons will pass through the particular electron transport system and produces the ATP, where in the inner membrane of mitochondria. So in the inner membrane of the mitochondria, the whatever the NADH plus or FADH which are produced in the matrix will be oxidized and releases the electrons, and those electrons will transported in the particular way through the four complexes and produces ATP. All this entire thing will be taken place in inner membrane, that is yours in the 19th. where it will be taken electron transport system. That is, every point is important for the need, remember it. So the two events, whenever it is taking place, the second event, whatever we told, electron transport, the electrons where it will reach, finally oxygen it will reach and forms the water. Here what we told, that it carbon dioxide and water also produced. Where the water is producing? In the electron transport system. This is a neat question. Why I am telling you this much detailedly? All are neat questions. Water is produced in the electron transport system. Carbon dioxide will be released in the matrix. How many carbons are there? 3 carbons. 3 carbon dioxides will be released in the matrix. All are the neat questions only. And next, this what it will do here, he has given electron transport system, water and oxygen, and ATP will be produced. Okay. Now pyruvic acid, there he has given one equation. That is pyruvic acid coenzyme A plus NAD plus in the presence of the magnesium. Remember this one properly, because magnesium he didn't mention in the text, but in the equation he has given. Such type of things only he used to catch in the NEET. So you have to see everything properly. So magnesium, in the presence of the magnesium, pyruvate dehydrogenase, this enzyme also you have to remember it. It produces acetyl coenzyme A, carbon dioxide, and NADH+. This is to happen in the first step of the before Krebs cycle. Krebs cycle, all of you know. That is this step. So this, after this one, when it will, where it will taken in the mitochondrial matrix. In the mitochondrial matrix, whatever is to take place, this is the Krebs cycle. But he has given heading tricarboxylic acid cycle, TCA, or the Krebs cycle or citric acid cycle. These two names he has given in the different places. Remember it. Heading he kept in the textbook, tricarboxylic acid cycle. Why it is tricarboxylic acid? The citric acid used to have 3 carboxyl group, functional carboxylic groups. For that only it is called as a tricarboxylic acid. And it is to cycle will take place based on the first product of the carbon of this whatever the pyruvic acid compound, whatever is coming, that is fixed and forms first formed in the Krebs cycle product is that is the citric acid. So it is called as a citric acid cycle or tricarboxylic acid cycle or Krebs cycle. All the names he has given in the different places, but the heading is given tricarboxylic acid cycle. Why it is tricarboxylic? Because it is having 3 functional 3 carboxylic groups, the citric acid. So tricarboxylic acid. In the tricarboxylic acid, in the textbook he has given, in under that whatever the cycle he has given, citric acid cycle he has given. But actual citric acid cycle is this one only. This is not, this is the before citric acid it is to take place. Remember it. There you will get the confusion. Means before entering into the Krebs cycle, the pyruvic acid will take some modifications and releases something. So then only the released product will come and enter into the Krebs cycle or citric acid cycle. Now you see, for that only, generally we used to call it as link reaction. In the matrix, whatever the pyruvic acid undergoes some reactions and finally forms the acetyl coenzyme A, which is to directly join in the citric acid cycle or Krebs cycle or TCA cycle, will be made. That entire reaction, connecting reactions together is called as link reaction. It is not in the textbook. If you remember that one, in this way, it will be easy to you. Now, what happens in the link reaction? Pyruvic acid, which is the 3 carbon compound, will undergo one reaction, complex, that is one reaction with the help of complex enzymes, complex of enzymes. The complex enzymes used to form pyruvic dehydrogenase. Remember, it is a complex enzyme, means it is not that much all the different enzymes, but it will be called as a pyruvic dehydrogenase. And here, for two coenzymes also will work in this reaction. They are the coenzyme A and NAD plus. Both are the coenzymes. This is also important point. It is given in the textbook. We can ask in the NEET. So this reaction, how it will be? Here pyruvic acid will be oxidized. Which is oxidizing? Pyruvic acid. First oxidation in the mitochondria is, he will ask, that is the pyruvic acid convert to the acetyl coenzyme A is the first oxidation it will do. In the first oxidation means releasing of the energy, and oxidation means sending out the hydrogen, or oxidation means sending out the electron. So it is to send out hydrogens, and these hydrogens will be taken by the NAD plus, and it is to reduce NAD into the NADH plus H plus. So first NADH plus is produced in the matrix is anything you can ask. Please remember the thing. Practice the cycle 4-5 times, it will come. Don't worry. And at the same time, it is to take the coenzyme A also in this reaction, and it releases the carbon dioxide. One carbon it loses in the form of carbon dioxide. So oxidative decarboxylation we used to call, because pyruvic acid is oxidized and carbon dioxide is coming out. Oxidative and decarboxylation and pyruvate dehydrogenase. In all the names, please remember. Otherwise, after this video, write it on the paper and read it properly. What is decarboxylation? NAD, so pyruvic dehydrogenase. Pyruvic is oxidizing. That is also important. So coenzyme A it has taken. Finally, it is to form acetyl coenzyme A, which is acetyl group is a 2-carbon compound. Acetyl group is a 2-carbon compound. This acetyl coenzyme means pyruvic acid 3-carbon compound lost 1 carbon and become 2-carbon compound. That is the acetyl coenzyme. The acetyl group is a 2-carbon compound. Now, this acetyl group will join with the citric acid cycle or the Krebs cycle or TCA cycle. This is the from acetyl coenzyme onwards, whatever the reactions, cyclic reactions are coming, they are called as a Krebs cycle, TCA cycle or citric acid cycle. Now, acetyl coenzyme A, how it is formed? Pyruvic acid is oxidized and NAD is reduced, carbon dioxide released, coenzyme A is joined into this and formed acetyl coenzyme A. Now, in this reaction, which is to receive the acetyl coenzyme A, oxaloacetic acid, which is 4-carbon compound. Remember this thing, 4-carbon compound. Acetyl group is the 2-carbon compound. This 4-carbon and plus these 2-carbon will become 6-carbon compound and forms the citric acid, which is 6-carbon compound. 4-carbon are the oxaloacetic acid, 2 carbons comes from the acetyl group and forms the citric acid, and this reaction will be done by citrate synthetase. This also given in the textbook, we have to remember it. And this reaction will be done by taking off one water molecule. This is also very, very important. I will tell you. This is given in the text but not shown in the cycle. So you draw this cycle from the video and practice it. Whatever the names given in the text and cycle, everything I covered in this one. If you practice this thing, it will become easy to you to follow the aerobic respiration. That is in the matrix, whatever the Krebs cycle has given. We having so many norms, that is complicated. For that one, some people used to do something. It is not better to remember it by practice. Then only whenever you are practicing, all the things, whatever the reactions that will come into your mind. If you only first letter by that one by singing and all things you do, you won't get. Practice it sincerely. When it is a very difficult one, why you want to spend 1 or 2 hour, 1 hour? 1 hour you can practice it, no problem. Next day half an hour, next day 15 minutes. Then we have to see 5 minutes for 4 5 days. Over weekly once if you refer, you can remember all the things. And one of the great thing is the questions and answers will be given on the paper of the NEET. So you can easily select the statements gives also you can understand, and the objective type also you can, and the matching also you can. But the only thing is by the practicing, not by the singing or the other thing. First letter, what is the names, not by hearting. By practicing only you will get remember it. So acetyl coenzyme A, acetyl group contains of 2 carbons, oxaloacetic contains of 4 carbons, and both they will be condensation will be taken place, and they will both will be condensed and forms a citric acid which is 6 carbon compound, and for this one citric synthetase will be used. That whatever blue color I given, they are given in the text but not in the cycle, remember it. For that only I am telling this, if you practice it is very okay. So for that one I given blue color. Citric synthetase is to convert oxaloacetic acid by condensing with the acetyl coenzyme A, and acetyl group with the oxaloacetic acid and form citric acid. Coenzyme A will be released. So acetyl coenzyme is what I told, it is the coenzyme, it will be released. So acetyl group only will be condensed. Not total acetyl coenzyme A. Remember this whatever I am telling, all are the neat points. You can ask that. So citric acid, which is a 6 carbon compound, will be isomerized and forms the isocitric acid. This also given in the text, blue color I written. Remember it. And isocitric acid undergoes two successive reactions and forms the alpha ketoglutarate or glutaric acid, both are the same. Pyruvate or pyruvic acid, both are the same, because pyruvate can be converted into the pyruvic acid very easily. Any ate it is coming, it can be converted into the acid very easily. So in the textbook also, if you observe, he will give in the text pyruvic acid, and in the diagram he will say pyruvate, or pyruvate he will tell in there he will give the pyruvic acid. So pyruvate or pyruvic acid or alpha ketoglutarate or glutaric acid, all are the same. That you should remember, that you keep in your mind. So isocitric acid will be oxidized and forms the oxalocitric acid. It is not present in the text, but for your convenience I wrote. This is the only step I given. So oxalosuccinate 6 carbon compound. This is not necessary to remember. But here what happens, isocitric acid converting into the oxalosuccinate, one oxidation will be taken place. And so, here some people used to write the enzyme names also. Isocitrate dehydrogenase means for this one dehydrogenate, for this one dehydrogenate, like that it is. It is not necessary, it is not the textbook, it is not necessary also. Only this enzyme only has given. So first oxidation will be taken place when isocitric acid is forming. Next 6 carbon compound oxalosuccinate, one NAD plus will be converted into NADH plus H plus. This is the first oxidation in the Krebs cycle. First oxidation in the matrix, this one. In the Krebs cycle, first oxidation. Like that also you have to remember. If you practice it, will be good. Now oxalosuccinate will be converted into the alpha ketoglutarate or glutaric acid, which is 5-carbon compound, means here oxosuccinate, whenever it is doing decarboxylation, whenever it is taking, one carbon will be sent out from the pyruvate, whatever it is coming, and one carbon it loses. So 6-carbon compound will become 5-carbon compound. This is also important for it. This entire Krebs cycle, 6-carbon compounds are here. 4-carbon forms are present here. This is the only 5-carbon compound which is to form in the Krebs cycle. Remember it. And by losing of one carbon in the form of carbon dioxide, the oxalosuccinate will become alpha ketoglutarate acid or glutarate, 5-carbon compound. Now, again, alpha ketoglutarate acid will be oxidized and decarboxylase like these reactions. It is taking here whatever coenzyme A taken, it releases the carbon dioxide. It is to, if you remember this one, this is the same. Like that you have to remember. Here whatever you done, whatever incoming coenzyme A, outgoing carbon dioxide, and NAD is it becomes NADH+. That's all. By practicing that one, it will become very easily. So alpha ketoglutarate will become succinyl coenzyme A because it has taken here acetyl coenzyme A, here succinyl coenzyme A is to form. That is the 4-carbon compound. Means here one carbon dioxide is losing. That is the decarboxylase, dehydrogenase, oxidase. All the things used to happen here. I told here no decarboxylase, oxidase, and it is dehydrogenase. All the things also will be taken place here. Now, this is the here one NAD forming the NADH+. This is the second oxidation. First oxidation here, second oxidation here. So, carbon dioxide released here and here. One carbon has gone, one carbon has gone. How many carbons are entered? 2 carbon. 2 carbons has gone over. So 4-carbon compound is formed. 2 carbons entered and they left the cycle in the form of carbon dioxide at the two places from the oxaloacetic acid when it is converting into the before alpha ketoglutarate acid, after alpha ketoglutarate acid, 2 carbons will lose. Like that also you can remember. It is easy. Before and after alpha ketoglutarate formation, before and after next product also 2 carbons will be left. And now here succinyl coenzyme A, this is not given in the cycle but wrote in the given in the textbook. In the he has given. So succinyl coenzyme A, which regenerates the oxaloacetic acid. So how it will be? This succinyl coenzyme A, because it has taken coenzyme A, coenzyme A will be released and it forms the succinic acid and energy will be released in this reaction. That energy, whatever it comes, it forms GDP to the GTP. GDP means adenosine, this will be GDP, guanine, dinucleotide. Guanine dinucleotide phosphate. Guanosine dinucleate, ATP adenosine triphosphate, diphosphate. Guanosine diphosphate will be converted into the guanosine triphosphate. So this guanosine triphosphate, adenosine triphosphate, adenine and guanine are the purines in the DNA formation, that you know, nucleic acid formation. Adenine diphosphate, adenosine triphosphate or guanosine triphosphate are both are the same. This will come in the adenosine diphosphate, guanosine triphosphate, thymidine diphosphate and uridine triphosphate, thymidine triphosphate, all will come in the nucleic acids in the molecular basis of inheritance. Okay, the GDP, whatever the guanosine diphosphate, it will be converted into the guanosine triphosphate by taking one inorganic phosphate. That also you have to remember, because he shown in this equation in the textbook. Because it is not taking the phosphate from the sectional coenzyme. May I remember it? So in that, he clearly mentioned in the equation. This type of things only will catch in the NEET. So by taking plus, I will write here, plus inorganic phosphate. Guanosine diphosphate will take the one inorganic phosphate and converts into the guanosine triphosphate. So here the and sectional coenzyme, a coenzyme will be it will go out like this, and it will become succinic acid, which is 4-carbon compound. So sectional coenzyme converts to the succinic acid by decarboxylation, by the giving out one GDP to GTP, and the guanosine triphosphate immediately it will converted into the ATP. And one more is there, in the animals GTP will form, in the plants ATP will form. Further than this fellow has given in the textbook, formation of the ATP has given, not GTP. Means immediately it will convert into the ATP or directly ATP. It is not necessary to you. It will GDP, guanosine diphosphate, will convert into the guanosine triphosphate by taking one inorganic phosphate. When succinyl coenzyme is converting to the succinic acid. That is the thing. 4-carbon. Now the succinic acid will be converted into the fumarate. The fumarate name also he has not given the cycle, but fumarate name given in the ETS, electron transport system. Succinic acid to fumarate he has given in the second chamber, second complex. There, so he has given, he can ask. So when the fumarate succinic acid, when fumarate it is forming 4-carbon compound, one more oxidation will be taken place. That is, FAD plus will become FADH2. Flavine adenine dinucleotide will become flavine adenine dinucleotide hydrogen. Here you should remember it is H2, not H plus H. That reason I will tell you in the electron transport. So FAD will be converted into the FADH2 when succinic acid is converting to the fumaric acid. And fumaric acid will be converted to the malic acid by taking the water. This he has not given in the text also. And but he has given here in the equation. So I am showing the two water molecules here and here very clearly, because he can ask. It is our duty to explain it. Where the two water molecules are used means one water molecule is used when the condensation of the oxaloacetic acid with acetyl coenzyme A, there one water molecule, and fumaric acid, fumarate is converting the malic acid, one more water molecule will be taken. And malic acid will be converted into oxaloacetic acid by one oxidation. That is NADH will be produced, NADH. Oxaloacetic acid will be converted into the malic acid. Where you learn? The C4 plants. Oxaloacetic acid will be converted to the malic acid, will enter into the bundle sheath cells study, that if you remember, this two are easy. Malic acid will be converted to the oxaloacetic acid. There oxaloacetic acid will be converted to malic acid. That's all. Like that you have to practice. With this you will study two things. What happens in the C4, and the same thing malic acid converts to oxaloacetic acid. So these two has came. And this reaction and these reactions are almost similar. Only few you has to practice it. That's all. Like that, if you practice and things, it is very easy. So now one more oxidation will be taken place. NAD plus NADH plus H. You people that I will see about this one I discussed in the C4 plants. Oxaloacetic acid converts the malic acid. It will bring NADH plus H also. That I discussed there, it is not necessary to you, because here it is giving out, when it is converting, it is taking inside. That is the reason. So if you give the same question, you will feel it as out of syllabus. But here, malic acid, when converting the oxaloacetic acid, it is oxidizing and producing NADH plus H. NADH, NAD or NADP, almost similar. DP will be the directly adding. NADH means the hydrogen will be separated in the ETS. Tomorrow I will discuss. So oxaloacetic acid will form. This oxaloacetic acid again will re-enter and cycle will be continued. Now in the matrix, how many oxidations are taking place? You will ask. First oxidation here, okay, one oxidation will be here, and 4 oxidations in Krebs cycle. First oxidation, second oxidation, and third oxidation, and fourth oxidation. In these 4 oxidations, one pyruvic acid is forming from in the matrix one NAD plus NADH plus 2, 3, and 4. For that only, in the has given one equation at the down of that citric acid cycle, that is pyruvic acid 4 NAD, here 1 and 2, 3 and 4. In the matrix he has given, remember it, this equation is for the matrix, mitochondrial matrix. And FAD, one FAD is 2 H2O, one H2O here and one H2O here. And ADP means here he has given ADP but GTP. ADP, GTP both are the same. For that one I am telling. And phosphate, inorganic phosphate. In the mitochondrial matrix, it used to give 3 carbon dioxides. 3 carbons will lose in the form of the 3 carbon dioxide. 1 will be in the link reaction, 2 will be in the Krebs cycle. Remember it, this is most important, never need. He asked also once. So 3 and 4 NADH plus are formed because 1, 2, 3, 4. And 1 FADH2 is formed here when the succinic acid is converted to fumarate. And 1 ATP means that is the GTP. ATP is formed. So this is from 1 pyruvic acid. Now in the next paragraph what he wrote, if it is the glucose, what happens into 2? Means they will 2 pyruvic acid for 8 NAD, all they will be double. So 6 carbon dioxides will come in the from glucose, 8 NADH plus form, 2 FADH2 is formed, 2 ATP are formed. That's all. In the next paragraph, that only he wrote. 1 pyruvic acid from glucose means you have to multiply 2 because 2 pyruvic acids are entering. That is the total product. Now, whatever you observed in the mitochondrial matrix, the reactions are taken as the link reaction and Krebs cycle. There is no utilization. We are keeping the heading aerobic respiration in the presence of the oxygen, but oxygen is not used anywhere. That oxygen will be used in the second event. That whatever the electrons removed, hydrogens pass through the ETS electron transport system and produces water, and in the with the oxygen. Oxygen is required in the electron transport system. That is the second stage. So oxygen, this also you wrote in the textbook, for that one I am telling. The thing is, here you see, once more I am explaining, stepwise complete oxidation in the eukaryotic takes place in the mitochondria to release large energy carbon dioxide and water. That is the aerobic respiration. And next, the transport from transport, the transported pyruvate means pyruvic acid from cytosol to the mitochondria takes crucial events. 2 events he has given. First event, complete oxidation of the pyruvic acid stepwise releases hydrogens and carbon dioxide. Carbon dioxide means carbons will be released in the form of carbon dioxides and produce NADH plus, FADH2, ATP in matrix it is to take place. All whatever I briefly I wrote, all are the neat bits. And the electrons removed from this one, it is this thing. In the where it is ATP in the inner membrane of mitochondria. Inner membrane used to form cristae. The cristae foldings and entire inner membrane contents of this one ETS. I will discuss that. That he has given one equation, pyruvic acid coenzyme A, NAD. That is the means link reaction he has mentioned there. That is pyruvic acid with coenzyme A and NAD plus with the presence of the magnesium. This you have to remember. Pyruvate and dehydrogenase, it will be acetyl coenzyme A, carbon dioxide. He has given only here magnesium. Some people what they will do, here manganese, this one, and enzyme name, all are not necessary. Whatever he has given in the textbook, you have to follow it. That much one. Beyond that one, he won't go in the it. So here you see that pyruvic acid, whatever it is converting acetyl group, that he has given in this equation. Now you see tricarboxylic acid cycle, TCA cycle, or Krebs cycle, or citric acid. Why did I discuss? Pyruvic acid will be decarboxylase, oxidase, and dehydrogenase. 3 types of the reactions will be taken with the complex of the enzymes. And 2 coenzymes. What are the coenzymes he has given in the textbook? He will ask definitely. Coenzyme A and NAD are the coenzymes. And acetyl coenzyme used to form. In the acetyl coenzyme A, acetyl group, which is the 2 carbon compound, will be condensation will be taken with oxaloacetic acid in the presence of the water. Water it will take in the presence of the citric synthetase enzyme. Citric acid will be formed, which is the 6 carbon, because the 4 carbon and this is the 2 carbon, it will become 6 carbon. And this is citric acid, immediately it will be isomerized. Isocitric acid will be formed. And isocitric acid will be oxalate succinate, it will form. It is not in the textbook, try to remember it. And first oxidation will be taken place. NAD will become NADH plus. And oxalosuccinate will converted into the alpha ketoglutarate or glutaric acid, which is 5 carbon. By lose it forms because of the losing of 1 carbon dioxide. And next alpha ketoglutarate, again it will lose 1 more carbon dioxide when it is converting into the succinyl coenzyme A. This reaction will be almost similar to this reaction. So here one alpha ketoglutarate will be oxidized, NAD will be reduced, coenzyme will be taken, and decarboxylation will be taken like this. And succinyl coenzyme A will be converted to succinic acid by losing coenzyme A, and the energy produced will be doing, that is the GDP to the GTP or the ATP, immediately it will form. And succinic acid 4 carbon compound will be oxidized again and forms the fumarate. So this 2 has given in the electron transport system. So here it is important, there also it is important. And these 2 are given in the C4 cycle. So succinic acid 4 carbon will become fumarate by oxidation and produce here FAD, flavyl adenine dinucleotide will become flavyl adenine dinucleotide with hydrogen. Fumarate will form, and this fumarate will be converted into the malic acid by taking 1 water molecule, and malic acid will be converted into oxaloacetic acid by oxidation. NAD and NADH will be produced. This is the 4th oxidation. Total oxidations are how many are taken place in the matrix? That is the here 1, and in this one 4. 3 are the NADH and 1 is the FAD. How many DTP or ATP are produced? Only 1 it is produced. And oxygen will be not used in this entire thing, but oxygen will be used in the next stage, that is the electron transport system. So this is the entire thing about the Krebs cycle and the link reaction which he has given. So complete oxidation will be taken place in the matrix, but releasing of the electrons from the hydrogen, that also oxidation. Two more steps will be there in the electron transport system. That I will explain you in the next video. It may come tomorrow or day after tomorrow. Now the thing is, here if you like this video, please subscribe the channel to see all the things. Now already I completed photosynthesis in this way. And now this whatever the respiration in plants, I completed. One more video is there. Electron transport system and balance sheet, and that is the amphibolic pathway, and next is that is RQ, respiratory coefficient. All the things are small things. One video it is enough. After that one, I will start the growth, plant growth and regular plant growth and differentiation and growth. That I will see. Later on, I will start the second year syllabus, which is that is the principles of inheritance, and next is the molecular basis of inheritance and principles of biotechnology. Why I chose these topics which are very difficult? All other things I will explain, but afterwards I will explain. This whatever I told, I am going to discuss all the things by the January ending. And whatever this I am discussing, minimum 20 bits will come. 20. Remember it. And most this only topics only will decide our medical seat. For that only I chose these topics and I am explaining. If you want to see all the videos whatever I am releasing, please subscribe my channel. And if you like this video, please give the like mark also to this. Okay, thank you.